Okay, this is where we left off last time, and I felt like at the end of the last lecture, I got kind of hurried there, and I threw a whole bunch of new terms at you right as we were leaving. So I just wanted to go through some of these terms that we have that involving fungal hyphae. The septate, the aseptate hyphae, I think is pretty explanatory. This term senocytic, that refers to any kind of cell that is multinucleate. So because there's no cross walls, there's lots of nuclei kind of swimming around there within the cells there. Hyphae can also be considered vegetative or aerial. Okay, vegetative hyphae are obtaining nutrients. When those hyphal tips branch and fuse with one another, we get large masses of hyphae, and we call that mycelium. All right, so here's a picture of a fungal mycelium. That's from your book. I'm pretty sure this is not a scanning electron micrograph, especially given the units here. Okay, if this was 3 millimeters, this is something that's, I don't know, an inch across or so. So I think that's some kind of light microscopy, but you kind of get the idea, this huge mass of fungus or filaments, kind of a mold. Our mushrooms, for instance, can have mycelium as well, or they do have mycelium, but they're usually underground. Okay, and then aerial hyphae are reproductive. That's where aerial spores would originate. So I found another graphic that kind of illustrates some of these terms, because I assume that that's mostly pretty new, and I think this might be helpful. So here's a hyphae that's growing out of a spore. All right, so spores are the reproductive structure for fungi. Here's an aerial spore over here, and there's lots of different forms of aerial spores that we are not going to get into the reproduction, but for instance, when this structure matures, it's growing off of a vegetative hyphae down here. When this structure matures, then these spores are released. In this case, it may be some wind that hits it, and the individual spores will blow off, and wherever they land, then the hyphae are going to grow from that. Okay, so mycelium, that's a huge clump of hyphae, and then the senocytic hyphae, or aseptate, A meaning without septa in between, or septate hyphae. This is a term that we're going to get to shortly. This is a modified hyphae, so stay tuned for that. This is a little picture of yeast budding. We're going to talk about that next. All right, so I think this picture is just referring to, here's some aerial spores growing off a vegetative hyphae. All right, so that was kind of the terminology to do with filamentous fungi. The other form of fungi are the yeasts. They are small. We represent their morphology as being globular. So they're not perfectly round, but they are sort of roundish. Okay, and they are usually going to reproduce by budding. So here in this picture over here, here's a yeast, and then basically a small portion of the yeast kind of buds off like this, and usually detaches and becomes its own yeast cell. If that bud fails to detach, then we have a special term for that, and it's called a pseudohyphae. So that's what it looks like over here in this picture, that mostly you have, a lot of buds that aren't quite detaching. That's a strategy for those yeast to maybe be able to penetrate into some, smaller area, we think. So let's see, helps organism penetrate. between cells. Okay. And some fungi exhibit both yeast-like and mold-like growth. So we call that dimorphic growth. So both yeast-like and mold-like, or we'll say filamentous. So here's an example of, this is actually Candida, one of the most common kinds of, I guess you'd call it a yeast more often, but it can form both a yeast-like form and a filamentous form. Typically it's going to do one or the other, and typically which form it takes is going to be dependent on temperature. This is an organism that causes mycoses or disease. In humans, it causes actually quite a lot of things like athlete's foot, other things like an overgrowth in the mouth, referred to as thrush. Mostly things that are not particularly dangerous to humans. Which form does it take in the human body? That's not a straightforward question that I'm not even going to try to answer here. We do have a couple professors that particularly study Candida, Dr. Cleary and Dr. Thomas. So if you are particularly interested in molds or pathogenic fungi, Dr. Cleary teaches a class about different fungal diseases. You'll want to look that up. And just a few more terms. And just a few more terms to share with you considering fungal nutrition. So how do they get their nutrients? We mentioned this earlier, they're chemoheterotrophic. So troph means to feed. And then basically this means that they get their nutrients from organic sources. We're going to pick that term apart in the next chapter, chapter 6. But both are carbon and energy source from organic sources. That's most common for fungi. A kind of a subset of the chemoheterotrophs, many fungi are considered saprophytic. That means they absorb nutrients from dead sources. All right, so another way to say that is that they're saprobes. All right, but some are parasitic, and that means they're getting nutrients from living organisms. Okay, it's actually much easier just to absorb nutrients from dead sources, because any, say like a plant that was dead, their cell walls are starting to break down. It's easy to kind of penetrate in there. A living cell is quite another issue, because they have, defenses and, intact cell walls. So typically a fungi that's parasitic is going to have modified hyphae, kind of beefed up hyphae, and we call those haustoria. Okay, so there's another term. So here in this picture, they call it a haustorium. Again, it's just a modified hyphae here that can penetrate into a living cell. Okay, so any fungi that's parasitic, that's, again, living off at the expense of another organism, any kind of fungal disease, whether it's a plant or whether it's on humans, we call that a mycoses. I think I used that term earlier. Okay, so again, that's a fungal disease. Okay, so that's pretty much some basic information about fungi. Your book goes a little bit into asexual reproduction. I used to talk about the different kinds of spores. We usually classify fungi based on the kind of spores that they make. I'm not going to go into that. You can read more about it if you're interested. Some fungi also reproduce sexually as well. And again, we're just not going to get to that this semester. All right, so before we move into Chapter 6, we'll pause for a little review question here since you do have an exam coming up. So here's some different flagellated bacteria. Which one of these represents an amphitrachous flagellar arrangement? All right, so this is just, spitting out, remember in your terminology, this organism up here, that's paratrichous, flagella all the way around. Here's a monotrichous flagella, only one. Picture C, that's lophotrichous, a tuft of flagella. And D here, we've got a flagella at each end, so that's going to be our amphitrachous. Okay, also remember we reviewed the different morphologies of bacteria. Probably the most confusing to keep in mind is that difference between a spirochete and a spirillum. Which one do you think this is? They're both helical organisms. If you remember, the spirochete has that internal flagella called an axial filament. Okay, these flagella are external, so this must be a spirillum. Okay, so remember those are a little bit shorter, a little more rigid external flagella. All right, so good, let's move on. We've done part of Chapter 6 the part that talked about culturing organisms. We're going to circle back and finish up the rest of it. Your book obviously talks about metabolism before it talks about growth, which kind of makes sense. Like an organism can't grow until it has the energy to do that. But for whatever reason, we're going to push forward with Chapter 6. So we're going to talk about what kind of nutrients that bacteria need to grow, different macromolecules, kind of basic stuff there. We'll spend a little time about how the environment that a cell is in, how that impacts their growth, and that kind of goes hand in hand with what you'll be doing in lab next week. We're thinking about some things like temperature and oxygen and how that influences growth. We'll talk a little bit about math, and we're going to do really simple math, thinking about how we can predict bacterial numbers, as well as looking at stages of a growth curve for microbes. And then, of course, we have to think about if we wanted to count cells, what are some ways that we commonly do that? So, as you would expect, microbes, and particularly we're thinking about bacteria specifically, are going to need the same kind of nutrients that we do. Okay, so any nutrient that a bacteria needs in large amounts, we would refer to as a macronutrient. All right, so things like carbon, hydrogen, and oxygen, bacteria need those for lots of different cell structures. So we'll be kind of general there. And of course, they need nitrogen for amino acids. Really remember an amino group there, present in all amino acids, nucleic acids. Okay, nitrogenous bases are part of nucleic acids. So a cell needs lots of nitrogen as well. About 95% of a cell's dry weight is these four elements: carbon, hydrogen, oxygen, and nitrogen. Okay, so let's see, what else does a cell need in large amounts? Phosphate. Again, find lots of phosphate in nucleic acids. Okay, of course, membrane phospholipids. And of course, ATP, adenosine triphosphate, so lots of phosphate. We could probably add sulfur in here as well. Some amino acids have sulfur in here. Not sure why I left that one out originally. Potassium, magnesium, calcium, These are all, iron are all necessary for enzyme function. They may be cofactors, for instance, that are necessary in order for an enzyme to work. Iron here is in parentheses just because some sources consider that a micronutrient rather than a macronutrient. So not everybody agrees about what to call that. We can't agree that it is necessary. Micronutrients are no less necessary than macronutrients. Bacteria need them in much, much smaller quantities. So sometimes they're referred to as trace elements. Really, things like zinc, molybdenum, manganese, chromium, and there's more. Really just enough that you would get in, say, tap water would be enough for organisms. Mostly they play structural roles in enzymes. Again, as often as some kind of cofactor. Okay. And then some bacteria need growth factors. And again, there's a lot of variability about which growth factors an organism needs. Some bacteria can make their own growth factors out of inorganic materials. So that's something we cannot do. We cannot manufacture amino acids. A lot of bacteria can manufacture at least some of them. So just some examples, amino acids, NADH, that's an electron carrier we're going to talk a lot more about soon, various vitamins. Again, sometimes some organisms need those in their medium if we expect them to grow. And then we have some terminology that helps us understand how organisms get those nutrients they need. How do they acquire an energy source? How do they acquire a carbon source? We could also talk about where they get their electrons from, but I left that off because usually electrons aren't a limiting ingredient for the growth of an organism. This chart is helpful, but I think these terms are just inherently confusing anytime you slap two prefixes together. It's always just a little bit confusing. So the photo and the chemo prefixes, those refer to how organisms acquire their energy source, whether they get it from light, otherwise the sun, or whether they get it by the breaking of bonds in chemical compounds, meaning they are somehow eating or absorbing some kind of organic compound. And then the prefix over here on the left, auto and hetero, those refer to where an organism acquires its carbon source. Okay, is it coming from some organic compound? Hetero actually means other, so I guess that means other than the sun. Or is an organism getting its carbon basically from carbon dioxide? Okay, so remember, carbon dioxide, CO2, is not considered organic. Something's considered organic if it has carbon-hydrogen bonds. Okay, so again, even though CO2 has carbon in it, it's not considered organic. So when we try to piece together these prefixes, we've talked about how fungi, a lot of protozoa are chemoheterotrophs. So that means they get their energy up here from the breaking of chemical bonds and their carbon source from organic compounds. So basically they're eating stuff, all right? This is very popular. We do this too. The other popular category is the photoautotrophs. So these are photosynthetic organisms. They're getting their energy from the sun and their carbon dioxide from carbon dioxide. And their carbon from carbon dioxide. All right, so anything that photosynthesizes, that could be a protozoa, it could be a bacteria, archaea, algae, right? All sorts of things. These other two categories are going to be very kind of rare, obscure archaea or bacteria. They're going to be environmental organisms. So if we went more into environmentals in this course, we would talk about that a little bit more, but any example that I would give you would be kind of foreign sounding. So we're not going to really worry about any examples there, but just know that there are some other, options out there, organisms that do some crazy things metabolically. All right, so let's look at some environmental influences that affect bacterial growth. Usually the first thing that we think of in terms of environmental influence is going to be the temperature. Because oftentimes, we have some degree of control over that. If we're trying to grow organisms in the lab, we can dial in whatever temperature we want. If we're trying to grow them fast, then we need to know what temperature they grow best at. Okay, so most organisms are going to have, some kind of range of temperature as to what they grow best at. So, and typically a little bit higher than their optimal temperature pretty quickly after that is too hot. Their enzymes start to denature and they're not going to be able to survive. Okay, so usually there's a little bit of a range. Here's a picture from your book that has E. coli. Same amount of E. coli was inoculated onto 3 different plates, and then those plates were incubated at different temperatures. One was left at room temperature, that's about 22. A little warmer than room temperature was this plate, and then this plate was incubated at body temperature, 37 degrees Celsius. So you can see E. coli grows the best at 37. That makes sense. It's an organism that lives normally in our body. Still pretty good at 30, not great at 22. All right, so still grows, just grows a lot more slowly. All right. And of course, different organisms are going to grow best at different temperatures, and we have terminology to describe the optimal temperature that an organism wants to grow the best. A bacteria that's considered a psychrophile grows best, typically we say somewhere below room temperature, so about 20 or maybe 15 degrees Celsius and below. Okay, cold-loving organisms. All right, so we have grown psychrophiles in the microlab in Henry. Even when you put them at that temperature that they like, they grow really slowly. So we lose interest after a while. These are going to be organisms that might grow, say, normally on polar ice caps. So kind of unusual places like that. They would grow really well in the refrigerator. Most of the organisms that we deal with are mesophiles, so they're growing somewhere between about 20 and 40 degrees Celsius, somewhere between room temperature and body temperature. Any organism that causes disease in humans is going to be a mesophile. Okay, so typically they're going to have their optimal temperature here about body temperature. And then thermophiles are our heat-loving organisms. You see different definitions of what the actual temperature is, but somewhere between about 45 and maybe 80 or so is a thermophile. And then our hypothermophiles, these are just crazy organisms. They're growing in like hot springs, even can survive at boiling. So that's pretty astonishing. So yeah, hot springs at Yellowstone, places like that. Your hot water heater at home even may have some hypothermophiles. And there's definitely some nuances here, like we have terms to describe an organism that grows best at body temperature, but also can survive lower, or there's again some nuances that we won't really worry about. All right, in terms of oxygen requirement, this is another thing that you're going to explore in lab next week. And I mentioned this test before when I was talking about anaerobic culture medium. Let's look at it a little bit more closely now. The thioglycolate test, this is a medium that has thioglycolate in it. This is a reducing agent that reduces any oxygen in the medium down to water. So with any broth test tube, you're going to have less oxygen in the bottom of the broth than what there would be at the top. But this essentially chemically gets rid of the oxygen. There's a loose fitting cap on there, so we're going to have some oxygen diffusing underneath that. And we typically have some kind of indicator that tells us, oh, this is the zone where we have about atmospheric oxygen. So we inoculate these, with a loop, stir it around, incubate it overnight. Then we look to see where the organism grows and try to discern their oxygen requirement based on where the organism grows. So if we've got growth here just at the top of the tube where there's oxygen, we would call that an obligate aerobe, an organism that needs oxygen for its metabolism. Okay. Conversely, if we have an obligate anaerobe, that's an organism that does not survive in the presence of oxygen. A facultative anaerobe is one with very flexible metabolically. They are going to be able to respire with oxygen, and they typically would prefer that because, as we'll see as we get into our unit on metabolism, they're going to get a lot more energy when they respire using oxygen as a final electron acceptor. But they are also able to either respire anaerobically, and that means, we'll talk more about this later, that they're going to use something other than oxygen as a final electron acceptor. Or maybe they're able to do some kind of fermentation. And we'll talk, again, what the difference is between anaerobic respiration and fermentation. But you're going to have growth throughout the tube, probably a little bit heavier here at the top because they're growing faster in the presence of oxygen. But again, facultative means flexible. So very flexible with their metabolism. And then you could have an organism that's considered an aerotolerant anaerobe. And in reality, it's kind of hard to tell the difference between these two just by looking for the growth, because it's, the theory is there, but in practice, which is kind of hard to tell. But an aerotolerant anaerobe is one that does not use oxygen, but is not poisoned by it. Okay, so for instance, the Streptococcus, most of the Streptococcus, all they do is fermentation. They don't really care whether they're around oxygen or not because they don't use it. But so they're just kind of doing their own thing, fermenting, and it doesn't really matter how much oxygen is in the tube. So we get kind of even growth throughout the whole thing. To add another nuance to this, it's actually not atmospheric oxygen that would, say, poison organisms. So the reason here that an obligate anaerobe is not growing here is not because they're poisoned by oxygen per se. It's actually other forms of oxygen that they don't have the capability to detoxify. All right, so I want to think a little bit about toxic forms of oxygen. Your book does address this a little bit. So here's 4 toxic forms of oxygen. I want to think a little bit about where these toxic forms of oxygen come from, why they're so bad for cells, and what cells, what do they have to do to survive in the presence of these toxic forms of oxygen. And I'm not worried about that you know the chemical formula for these. I just kind of bop that in there. Singlet oxygen, this actually is formed as a byproduct during photosynthesis. It's when oxygen is actually boosted to a higher energy state. we don't most any kind of organism that's not photosynthetic wouldn't really encounter that. Plants have pigments like carotenoids to counteract. So that's not something that, again, most bacteria would have to deal with. These two, superoxide and peroxide, those are formed as a byproduct during aerobic respiration. Okay, so same thing formed during aerobic respiration. So those are all over the place. Okay, lots of organisms that are respiring aerobically. Those have to be dealt with. And hydroxyl radical, this one is not as common. It's probably the most toxic because it has an unpaired electron here that's grabbing electrons from other molecules. This is formed as a result of ionizing radiation, so that's not going to be commonly encountered either. So it's kind of these two, superoxide and peroxide anion, that cells have to deal with. What's so bad about all these different forms of oxygen? Well, they're all strong oxidizing agents. And that just means that they are grabbing electrons from other molecules. Okay, so when they tear off those electrons from other molecules, they're causing just all kinds of damage in the cell. Okay, so organisms have to have a way to deal with especially the superoxide and peroxide anion. Okay, so they do that by being able to synthesize an enzyme that detoxifies them. All right, so for instance, again, don't memorize these equations, but there's an enzyme called superoxide dismutase. That's any organism that lives, in the presence of oxygen, or any organism that respires aerobically has to be able to produce superoxide dismutase to detoxify superoxide into water. And, not water, hydrogen peroxide and oxygen. Okay, now the problem is this hydrogen peroxide, this is also toxic to cells. Okay, that's the peroxide anion is part of hydrogen peroxide. So they also need to have some enzyme to detoxify hydrogen peroxide. Okay, and one of those enzymes is catalase, detoxified into just water and oxygen. Okay, and this is going to be a fairly, let me just outline this in another color here, the enzymes here. Okay, this is going to be a fairly familiar enzyme because this is one that you will be testing for in lab. This helps us tell the difference between certain organisms, whether they can produce catalase or not. So what you do, you rub your cells on a slide just dry. You take them from a plate, and then you add a drop of hydrogen peroxide. And if you see this vigorous bubbling, that is the oxygen being released. And so you know that organism is able to detoxify that hydrogen peroxide. Okay, that's how we tell the difference in the lab between Staphylococcus and Streptococcus. Staph can make catalase and Strep can't. Okay, Streptococcus actually use a little different enzyme to detoxify hydrogen peroxide. It's called peroxidase. And we get a little different end products, water in the electron carrier NAD. Okay, this is I don't even know how you would test for this. You have to have some way to either test for the substrate or the product to see if the organism's making the enzyme. And water in NAD, I don't know how you would test for that. But Basically, our streptococcus detoxify hydrogen peroxide by peroxidase, and our staphylococcus detoxify by catalase. And many organisms make catalase. We make catalase in our cells. Okay, so just again, very important to detoxify toxic forms of oxygen. If an organism doesn't have those detoxification enzymes, then they're going to be an anaerobe. Okay. That may be a little bit of a simplification, but that's what we're getting at. All right, most organisms can tolerate a little more narrow window of pH. That hydrogen ion concentration can interfere with bonding. But again, we've got terminology to describe bacteria as to what kind of pH that they grow best at. So an organism that's an acidophile will grow best at acidic pH. All right, so from the graph there, that's pretty straightforward. Neutrophiles grow best at neutral pH. So most of our day-to-day organisms are going to be neutrophiles, E. coli, whatever. And then an alkalinophile or an alkylophile, both are correct terms, grow best at basic pH. Okay, so there are organisms that even grow at pH of 1. That's like, I think they make sulfuric acid, but places, mine runoffs, places like that are just crazy acidic. Some organisms, like even the organism that causes cholera, it actually grows best at about pH 9, a little alkaline pH. So again, there's just a lot of variety in the bacterial world. Okay, and let's explore a little bit the physical effects of water. So when we talk about osmotic pressure, okay, don't confuse that with air pressure. We're not talking about organisms that live, say, at the bottom of the ocean. We're talking about organisms that are able to live in a solution that has a very high solute concentration. Okay, so I think the easiest way to think about this is how badly water wants to leave the cell. Okay, so in a really hypertonic medium, okay, then water is going to leave cells. Okay, and that's typically, it's true that we could have a really hypotonic medium. We could have distilled water. Really, in nature, you're not going to have distilled water. So it's usually not a problem where too much water is coming into a bacterial cell, again, because of that cell wall. Usually the problem that cells are dealing with is when there's too much solute in the medium and then the cell is dehydrating. Okay, so you have a really hypertonic medium, then water is really going to want to leave the cell. Somebody said to me once, water follows stuff. Okay, so water is trying to, equal out the concentration of solute. Interestingly enough, there are bacteria that grow best in hypertonic mediums, and we call those osmophiles. So let's see, they grow best in high osmotic pressure. So some solute is in a very high concentration outside of the cell. We most talk about a particular kind of osmophile called a halophile. Okay, they are going to grow best in a high salt concentration. Okay, so these are not organisms that you find every day walking down the street. All right, these are extremely salty places. We're talking about the Dead Sea out in the Middle East. We're talking about the Great Salt Lake. where salt concentrations can be up to like 23, 24%. So extremely salty places, these organisms are not pathogenic. They are not, going to be able to grow in the body. We could have what we call a facultative halophile, where an organism doesn't grow best in high salt concentrations, but it can survive them to some extent. Okay, and this is what you're going to explore in lab. You're going to inoculate different organisms on increasingly hypertonic mediums and see, is there a difference? Do some organisms tolerate salt better than others? I think you'll find that they will. And lastly, I just want to draw your attention, I don't think the book talks about ecological associations, but we're more and more aware that organisms act differently when they are around other organisms than if they're in pure culture. So remember what a pure culture is. That's when an organism is in a petri dish or in a test tube, and there's only one organism in that petri dish or test tube. So that's why we like to study organisms when they're off by themselves, but we have to understand that they may act differently when they're not in their ecological niche, however you say that. Okay, so we can describe the association when organisms live with other species as either antagonistic, synergistic, or symbiotic. Okay, so an antagonistic relationship is when one organism benefits and one is harmed. Okay, so here's an example up here. We've got some kind of mold, this big white fluffy thing that's growing on some media. This bacteria that was growing there before now is suddenly inhibited. You've got this zone where the bacteria is not growing anymore. Okay, so that would be an antagonistic relationship. You can think of a ton of these, basically also known as parasitism. Okay, any organism that's living at the expense of another, a tapeworm living in our body, that's an antagonistic relationship. A synergistic relationship is where the members can live separately, but they benefit from the association or the relationship. Bacteria, we know, sometimes live in biofilms. So they sometimes live in community, and that seems to help them, but they can also, live separately by themselves. Dogs and humans, like dogs can live separate from humans, but I think both humans and dogs benefit when they live together. So think of some examples there. And then there are symbiotic relationships. Both organisms benefit, and one rarely lives without the other. Okay, basically they're not well adapted anymore to live separately. And a good example of that, there are bacteria that live in nodules on the roots of pea plants. Okay, the bacteria fix nitrogen into a usable form for the pea plants, something that's limiting in their environment, and then the bacteria gain lots of energy from the roots of the pea plant. Okay, there's also lots of bacteria that live in a termite's gut or a cow's gut. Okay, the bacteria get the nutrients from the cow or the termite, and in turn, the bacteria digest nutrients that the cow or the termite can't. All right, so lots of examples there. All right, let's take a closer look now at the growth of microorganisms from kind of a mathematical standpoint. We know that bacteria reproduce by binary fission. Not sure if I've used that term before, but basically one bacteria, a septum forms, usually, somewhere in the center of the cell, and one bacteria splits into two. Okay, it's asexual reproduction. All right, and we can describe the generation time as, try again, the time it takes for one bacteria, or we'll say bacterium, to become two. Okay, and we also refer to that as doubling time. All right, so if we kind of drew out how an organism splitting by binary fission, so up here we've got one bacteria, it gets a little bigger, a septum forms, and eventually it becomes two. All right, let's say that generation time is, say, 30 minutes. Like we can figure that out, how long that takes by watching them sometimes, or counting them. All right, and then eventually 2 cells become 4 cells. All right, there's another generation. 4 cells become 8. I don't know why they drew 9 cells here. So pretend that's 8. There's another generation. 8 cells become 16. So that's 1, 2, 3, 4 generations. All right. So we can quantify that by graphing that out. All right. So when we have bacteria populations doubling every generation, we describe that as logarithmic growth. And if we were to chart that out, we would have this kind of a parabola, that kind of a shaped graph like that. Very quickly becomes a lot of cells when it's growing exponentially like that. That logarithmically or exponentially, if you compare that to a species that's growing arithmetically, that just means that a population is adding a certain amount, like over a time frame. So for example, every generation the population is gaining like 2 organisms, right? So if that was the way a population was growing, you would see very slow growth over time. Okay, nothing like how the numbers increase with logarithmic growth. So species don't typically reproduce arithmetically. They may do it if there's something limiting their growth. So the carrying capacity of the ecosystem, it will only support a certain number of certain organisms. So their growth is going to be really slow over time. There's not enough food for, all the offspring to survive. Okay, so the generation time of any organism is going to depend on what kind of organism it is. In terms of bacteria, some bacteria will have a very short generation time. Like E. coli, their generation time is only about 20 minutes in an ideal environment, where some, like the Mycobacterium, their doubling time is more like 24 hours. So there's just a lot of variability depending on what species it is. Depends on the availability of nutrients. Okay, and it depends on lots of physical parameters, like the ones that we just talked about. For instance, temperature, pH, things like that. So just another word about growth curves. Typically, any population that it's growing exponentially, if we graph it on, like a regular plot where we have, time on the X and number of cells on the Y. When it's doubling every generation, like I said, you very quickly get a huge number of cells. And if you wanted to use this graph to extrapolate, well, how many cells am I going to have at 10 hours? Like, yeah, you're going to need a really big graph to try to match that up and to try to say, well, let's, to bring your, to extrapolate out from anything. It's not going to be terribly accurate. So typically when we're graphing numbers of bacteria, we like to use some semi-log paper. So that means time is kind of on a regular scale, and then the y-axis, the number of cells, we plot out on a logarithmic scale. So that way our numbers can get really big here, and our growth curve ends up being a straight line. So if we want to know how many cells we have at 10 hours, we can, drop a, draw a line over and figure out the number of cells that are likely to be there. So it's just a little bit more helpful, again, to graph using semi-log paper. And you may do that if you take an upper level microbiology class. Okay, so thinking about calculating cell numbers, again, one way to do it is extrapolating from a graph. But one equation that you can use, and I think maybe they have this in the book and perhaps not. I'm not going to go into this in too much detail, but if you want to calculate the final number of cells in a culture, if you know the initial number of cells that you inoculated in that culture, and N here stands for N equals number of generations. Okay, if you know how many generations and how many cells you put in there to begin with, so if you know the generation time, then you could calculate, your final number of cells. And, if any one of those variables is what you didn't know, you could figure that out. We are not going to worry about making a calculation like that. But if numbers aren't too big, you can also think through a kind of a guess of how many numbers you're going to have without using this equation. Okay, I'd like to have you try to think through this question without using that equation, and I'm going to show you how to do that. I think there's a question maybe similar to this in the homework, and I've posted one on your practice questions for the exam. Hopefully it wasn't the same question. I have to take a look at that again. Okay, so assume a culture of bacteria is growing exponentially. Right? If you have 20 cells in a broth culture and the generation time is 30 minutes, how long will it take until there are 1,000 cells? All right, so the way to think through this, it says you have 20 cells, and that's basically your time 0. Okay, that's what you're starting with. Okay, after one generation, which is after 30 minutes, how many organisms, how many cells are you going to have? Well, remember the population is going to double. So you're going to have 40 cells. All right. After a second generation, you're going to have 80 cells, and that would be an hour. All right. After 3 generations, again, this is going to double again, 160, hour, 30 minutes. Okay, you get the idea here. After 4 generations, you're going to have, 320 cells. That's going to be 2 hours. 5 generations. 640. 2 hours, 30 minutes. 6th generation, you're going to have 1,280, and that is 3 hours. So how long will it take until there are 1,000 cells? Your answer right there is going to be 3 hours. It could have asked, how many generations will that be? Okay, 6 generations. Okay, so again, I think there's a practice on your homework to figure out a question like that as well. The mistake that students usually make is they may count 20 as their first generation, and then they're going to be off with their count here. So keep track, that starting number, that's your time 0. Okay, and there are ways that terms that we like to use to describe the phases of microbial growth. And of course, this is going to be in a closed system. So in the lab, say in a test tube, we can describe that growth typically in 4 different stages. Okay, so right here, time 0, that's going to be when your culture is inoculated. You first introduce your cells, say, into your broth test tube. Okay, for a time, the cells are actually not growing very fast. We would call this the lag phase. Okay, here, that's when cells are adapting to their environment. Again, slow growth, not exponential. Okay, they may have to have produce certain enzymes in order to metabolize whatever nutrients they're given. Okay, but eventually they get kind of acclimated to their culture, and they are going to enter the exponential phase of growth, also called the log phase. Okay, this is going to be their maximum rate of growth. Cells are going to be very uniform in terms of their size and their shape. And actually, this phase, in the log phase, is when cells are going to be most susceptible to antibiotics. A lot of antibiotics don't work unless the cell is actively growing. Okay. Eventually, they will enter into a stationary phase. That just means that the number of new cells being produced are going to equal the number of dying cells. So it doesn't mean that all the cells just kind of sit there and stay the same. It's, again, number of news equal to the number of dying cells. So growth slows way down as typically the nutrients are being depleted. The waste products may be accumulating, and that may be hampering their growth. And eventually, they're going to enter either what we call a death phase or a decline phase, and cells are going to be dying faster than are produced. Okay. So, how do we know what stage a culture is in? Well, you have to know the numbers of bacteria in the culture, and not just at one point, but over time. And then you could make a growth curve like this. Okay, so we have to have ways to quantitate bacterial growth. And so I'm going to talk about a number of ways to do that. There are 2 different methods that require incubation. Okay, viable plate count is one. That's a technique that typically we do right away. It's the 3rd lab we typically do, but we put that off this semester. You're going to get to do it at the end of the semester if we have time. So I'll explain that method a little bit. Another method is with membrane filtration. Okay, so I'll come back to this slide in a minute. This slide explains this viable plate count method for estimating microbial numbers. So what this picture doesn't show a very good job of, it's got some test tube, and you're trying to figure out what we consider the original cell density, or we'll say OCD, basically the concentration of bacteria in some original sample. So you take an aliquot, a mil of that original culture, And typically we make serial dilutions here, because if we were to just put this culture right on a agar plate, we would always get way too many, almost always, assuming that this is a very concentrated culture. So we want to dilute that out, because we want to try to get kind of what we say a countable range of colonies to grow up between I'm blanking, 30 and 300. Okay. Basically, you get more than 300 colonies on a petri dish like this, and they're so close together you can't count them accurately. If you get less than 30, then it's not considered statistically significant. So you got to find that sweet spot where the numbers kind of mean something. And I'm not going to talk about like how to make dilutions and how to do the math. I'll leave that for your lab professor. Usually this is where we do a spread plate. Some of you did that in lab last week. You're not going to use a loop here. That wouldn't make any sense. Okay, so we would take this number, and taking into account the dilutions, we would be able to extrapolate back to this original sample. Okay, so we needed to culture bacteria in order to get those counts. Okay, another example where we're culturing bacteria is if you have some kind of original sample that doesn't have a lot of cells. So a sample with few cells. So most often this is going to be some kind of maybe a water sample where you would have to concentrate it, right, in order to get a countable number. So we would pour that water sample into some kind of vacuum system. There's a membrane right in here that we're going to run that water sample through. And typically we would have to suck that through with a vacuum in order to get the liquid to go through there. So we take that membrane off and put it on some culture medium here, and let it incubate, and then count the number of colonies. All right, so here's a real life picture of what that might look like. Okay, I've done this before with water from the Grand River, and it was totally unnecessary to concentrate it. Okay, there's a lot of cells, a lot of bacteria in the Grand River, and I could have just put that right on a plate, or maybe even diluted it. If you were to take a sample of drinking water, you would probably have to, put a measured amount through here, and then with the number of colonies that grow up, you could extrapolate back to about how many cells were per mil. Okay, so with both these methods that require incubation, what's the kind of pluses and the minuses of using these? Well, with both of them, kind of an advantage is that you are only counting live cells. Usually you only care about the living ones, so that's kind of an advantage. You're only counting, a colony is only going to arise from a living cell. So you're counting those colonies which represent a living cell. And with both of them, I would say the materials are pretty inexpensive. All right, you need some media, some flasks or whatever, some petri dishes, pretty inexpensive. So those are some positives. Some negatives with both of these methods that basically they take a little bit longer than other methods, because there's going to be at least an overnight incubation, depending on how quickly your cells grow up. All right, so it takes a little time to get that number. And again, you need to have counts that are statistically significant, so you might have to dilute or concentrate. With other methods, you kind of have to have that right number as well. Okay, so let's look at some other methods that don't require you to actually grow anything up. 4 different methods here. You could use a specialized glass slide called a Petroff-Hauser counter. Another name for this is a hemocytometer, and I typically call it that just because it's a little bit easier to say. But it looks like this. It's a big, thick glass slide, and you suspend your cells, in a broth or whatever medium, and typically you put a cover slip over it like this, and then the cells that you are trying to count, you put a drop like right here in this groove with a micropipetter perhaps, and then that suspension will wick underneath that cover slip, kind of an even suspension. And right here and here, there's actually a grid like this printed on that glass slide. You can't see that with the naked eye really, but when you look through the microscope, you can see that grid. And then you look at that even suspension of cells, and basically you count the number of cells in a certain number of squares there, and you can extrapolate back to how many cells are in your original sample. All right, so this is really a quick method. You do your counts and you get your numbers for how much is in your original sample right away. This method works a little bit better with eukaryotic cells like yeast, because you can tell very easily by looking at a yeast cell whether it's dead or alive. If it's dead, it's going to be all shriveled up. With bacteria, you can't tell as well whether it's dead or alive, and sometimes they're just small enough that it's kind of difficult to see. So, and again, you have to have kind of a countable range. If you only see 2 cells or whatever, it's not statistically significant. You get too many, you just can't count it accurately. So, but it is fast. You can use turbidity using a spectrophotometer. So if you took Bio 120, you should be familiar with a spectrophotometer. Remember, what that machine does is it shines light through a sample. And so you can quantitate basically the color of a sample, or in our case, we're trying to quantitate the turbidity of a sample, because sample, right? So and you would actually have to zero this out using some media that's sterile. Okay, so that's your zero, and then you can measure basically how cloudy or turbid your sample is. Okay, so that's going to give you an absorbance reading. Okay, that's not going to give you an actual number. But if you say, take your absorbance reading, it's 0.3, and then you count those cells using a hemocytometer, you can make a standard curve like this, and kind of know, if this absorbance, then I'm going to have, this number of cells. And then the next time you do your assay, you'll know, 0.3 is equal to a certain number of cells. Okay, so it doesn't give you actual numbers, but this is really nice method to know if your culture is actively growing. All right, take it out, test it after an hour. Look at the absorbance. Take it after 2 hours. Is the absorbance gone up or not? Okay. All right, and probably another drawback is that live and dead cells scatter light the same. All right, so you can't really tell if your cells are dead or alive from this sample, from this method either. Okay, getting a little bit more fancy, a little bit more expensive is using a Coulter counter, and we do have one of these in the lab that, again, I think the upper level microbiology labs use. This works on the principle that bacteria are going to disrupt an electric field as they pass through it. So basically you have your sample of bacteria here, and you pump it, it pumps through, and passes through electrodes, and when a cell passes through, that will basically show as a pulse. Okay, so you have to kind of get your flow rate just right so it's able to count those cells accurately. But it is probably a little bit more accurate way to count cells. It's good if you have a little bit, of a less dense population. And once again, it's kind of better for larger cells. Doesn't always work as well for bacteria. Bacteria tend to clump. So that's going to affect your numbers being a little bit less accurate. And again, you can't tell if they're dead or alive. So that's another little bit of a drawback. And we're getting a little bit more expensive than, using a spectrophotometer or just a hemocytometer. All right, and then one more method, we're getting top of the line now. It's a device called a flow cytometer. These, a basic model can be fairly inexpensive, maybe like $10,000, but you get one with all the bells and whistles in there, over $1 million. Okay, they use a light-sensitive detector that can actually separate out the cells once they've been stained with some kind of fluorescent dye or tagged with a fluorescent antibody. Okay, so we can not only label or stain different kinds of cells, but you can also then separate different populations out. Okay, it's very accurate. Okay, you can even tell living cells from the dead. Unfortunately, lots and lots of money. And the data can be a little bit more difficult to interpret. I know where I went to school, the researcher that I worked with, he from time to time would take some cell populations to the flow cytometer when he wanted to know he was looking at different cytokines and different kinds of immune cells, and he wanted to separate them out. We employed a full-time PhD to run the flow cytometer and help interpret the data. So it's complicated, but the data might look something like this. So I think this is some data I pulled from an issue of Science that it was different populations of cells in seawater. And like I said, you add a fluorescent dye that's going to stick to, different populations. So then you can see here, like the red dots, that's a particular kind of organism. The blue dots, that's some other kind of phytoplankton there. And then the green is, some other type of cells. So you can kind of see the relative numbers and even separate them out if that's something that's worthwhile. Okay, so that worked out just about right to be about a lecture worth. This is the end of the material that will be covered on exam 1. And thanks for listening.